Hello students, welcome to our channel Mootechie. This video is all about the phylogenetic tree in microbial classification. We're excited to share this foundational concept with you. Let's begin with the core concept. Concept of a phylogenetic tree. A tree is a hypothesis of evolutionary relationships shown as a branching diagram. Think of it as a family tree for species, proposing how they are all connected through evolution. It depicts clades, sister taxa, and patterns of descent. A clade is a complete branch representing an ancestor and all its descendants, helping us group organisms meaningfully. It can be rooted, showing the direction of time, or unrooted, showing only relationships. A rooted tree points back to a common ancestor, while an unrooted one just maps who is related to whom. Branch lengths may encode genetic change, topology shows relationships. Longer branches can mean more genetic differences, while the pattern of branches shows who split from whom. This visual tool is essential for understanding life's history. Meaning of phylogeny. Phylogeny equals the evolutionary history and relationships among organisms. It's the story of how life diversified from common ancestors over billions of years. In microbial biology, phylogeny is inferred from molecular sequences like 16S rRNA or core genes. Because microbes can look similar, we use their genetic blueprints to uncover their true family ties. It's important to distinguish phylogeny, which is about evolution, from taxonomy, which is about naming and ranks. Phylogeny tells us who is related, while taxonomy gives those groups official names and categories. Phylogeny captures common ancestry, divergence, and shared traits. By studying it, we can trace back to where lineages split and understand why certain traits are shared. Now, let's look at this illustration of the photogenic, or rather, the phylogenetic tree of life. We see the three domains again. Under bacteria, we have groups like green filamentous bacteria and spirochetes. Archaea contains halophiles and thermophiles. Eukarya encompasses familiar groups like animals, fungi, and plants, as well as microbial eukaryotes like ciliates and flagellates. This domain includes all organisms with complex cells containing a nucleus. This tree beautifully illustrates how all life on Earth is interconnected. Now, let's explore the different formats these trees can take. There are several types of phylogenetic trees. A cladogram shows branching order or topology only. Branch lengths are not scaled. It's a simple sketch of relationships useful for seeing who groups together without worrying about the amount of change. A dendrogram is a product of hierarchical clustering. It visualizes similarity-based groupings. Often used in data analysis, it clusters organisms based on how similar their traits or genes are. A phylogram has branch lengths proportional to evolutionary change, like the number of genetic substitutions. Here, the length of the branch matters, telling you how much evolution happened along that line. Trees can also be rooted, showing direction of time from a common ancestor, or unrooted, showing relationships only. Choosing a root gives the tree a timeline, while an unrooted tree simply maps the network of connections. Each type serves a different purpose depending on what scientists are asking. How are these trees built from modern genetic data? Let's discuss construction methods. Distance matrix methods build trees from pairwise distances between sequences using algorithms like UPGMA or neighbor joining. It's like building a tree based on a table of how different every organism is from every other organism. Maximum parsimony chooses the tree that requires the fewest character changes, emphasizing simplicity. It operates on the principle that the simplest explanation with the fewest evolutionary steps is often the best. Maximum likelihood finds the best tree under a specific statistical model of evolution, maximizing the probability of seeing the observed data. This is a more computationally intensive method that uses models to account for different rates of genetic change. Bayesian inference samples a posterior distribution over possible trees using Markov chain Monte Carlo and reports posterior probabilities for branches. This powerful method gives us a measure of statistical confidence in different parts of the proposed tree. Why does this matter for microbes? Let's cover importance in microbial classification. It refines microbial taxonomy by tying names directly to evolutionary history as seen in the three-domain system. This means our classification system reflects true biological relationships and not just superficial similarities. Phylogenetic trees enable pathogen identification, outbreak tracing, and antimicrobial resistance surveillance using whole genome sequencing. By comparing the family trees of bacteria, we can track the spread of a disease or a resistant gene in real time. These power key microbial metrics like UNIFRAC and phylogenetic diversity for comparing communities. These tools don't just count species, they measure how evolutionarily distinct the members of a community are. Trees also reveal events like horizontal gene transfer and support functional inference for unknown genes. If a gene sits on a certain branch of the tree, we can make educated guesses about its function based on its relatives. Now, looking at this illustration of the photogenic or rather the phylogenetic tree of life, we see the three domains again. Under bacteria, we have groups like green filamentous bacteria and spirochetes. Archaea contains halophiles and thermophiles. Eukarya encompasses familiar groups like animals, fungi, and plants, as well as microbial eukaryotes like ciliates and flagellates. This domain includes all organisms with complex cells containing a nucleus. This tree beautifully illustrates how all life on Earth is interconnected. Thank you from Mootechie for joining us on this journey through microbial phylogeny. We hope this exploration has illuminated how scientists use the family tree of life to organize, understand, and explore the invisible world of microbes. Your curiosity drives this discovery.